That's the first one. Okay, so yeah, so when I was discussing with Knut about the content of my talk, I realized that it's been 20 years since we first published a paper on the use of carbon-13 labeling to phenotype, to study the metabolism of Pichia. Okay, and actually was my first Pichia conference back in the same room we are now. In 2006, was the first time I presented this work to the audience. Okay, so as you heard from Dieter, carbon-13 isotopic tracer techniques can be used for metabolic pathway verification and even discovery. Also to assess metabolic network topologies, meaning to see which pathways are active or not in a given growth condition. But what we were particularly interested when we started this work was to use these techniques for metabolic phenotyping, meaning getting into to measure in vivo the metabolic rate reactions, the fluxes, what we call metabolic fluxes. Okay, so to do this, you need essentially metabolic model, so mathematical model describing the metabolic network, and then you need to go into the lab, okay, and perform some experiments to measure extracellular rates, reaction rates like consumption, substrate consumption rates, production rates, and on the same culture you need to feed cells then with a substrate that it's partially labeled with carbon-13, either uniformly on on every carbon or in some positions of the carbon source. Okay, so using these labeling patterns you get in the metabolites. These labeling patterns are a function of the stoichiometry of the network plus the reaction rate. So you can calculate from there your reaction rates. Again, and why it's so interesting, at least from our point of view, it's because reaction rates are actually the functional endpoints of multiple interactions across the different regulatory layers involving genes, proteins, and metabolites. Okay, so that's why we call it metabolic phenotype. Okay. Yep. So the idea behind that is that we were particularly interested in studying what was the impact of environmental perturbations, both environmental and genetic perturbations on the metabolic network. Okay, as a way of learning how Pichia responds, for instance, to different environmental changes on carbon source, growth rate, and oxygen availability, for instance, which are parameters that are relevant from the process point of view. Okay, and how these interact with what would be intrinsic stress factors like the metabolic burden caused by recombinant protein production or by the introduction of synthetic pathways, metabolic pathways, okay, different genomic additions, okay. So that was the interest behind. The classical workflow for conventional carbon-13 based metabolic flux analysis is very much well established. You need to start with to working under metabolic steady state conditions, meaning exponential growth phase or chemostat cultures, okay, that's one of the starting or the points you need there. Then you can perform experiments essentially. Let's see if I can. It doesn't matter. On the top picture, you can use either a substance that is uniformly labeled, so all the positions are carbon-13, or positional labeling in some of the carbons, and then you feed the cells with this substrate and then essentially measure labeling patterns in either protein bone, amino acids, or free metabolites, which is more experimentally demanding, but it can be done. Okay, and you do this at the isotopic steady state, meaning that you have to wait a long time to read the enrichment of carbon-13 reaches a steady state level. You can do it also transient, so measuring the kinetics of carbon-13 incorporation, but this is more experimentally demanding. It can be done. Okay, so then you can, once you have labeling patterns, you can recalculate, okay, metabolic fluxes by different ways. So one is this global iterative fitting of labeling at time fluxes. Okay, or you can also calculate local branch points, like what we call flex ratios. Okay, but the problem with methanol is that it's one carbon, so you need and for this type of experiments you need position-specific substrate labeling. So in one carbon, it's impossible to have position labeling. Okay. So how do you achieve this differential isotopic labeling during carbon one metabolism? One way to do it is to feed cells with both methanol or one carbon metabolite plus a multi-carbon source. Okay, we try that, very complex. Okay, the other way it's doing this transient labeling, so you feed just methanol and you measure the kinetics, okay, of incorporation of carbon-13. And the other one is that what just Didit said a few minutes ago, it's the inverse transient labeling. So you start with carbon-13 and you dilute, you introduce methanol unlabeled, so you trace that also in kinetics, okay? But when we started, we decided to investigate a new method for this for studying methanol metabolism, which is it's based on the uniformly labeled co-substrate. So both substrates, okay, so multi-carbon and methanol are uniformly labeled. So all carbons are carbon-13, okay, and this is what we developed in the early days. Okay, and it resulted quite powerful technique. It worked. Okay, so essentially you've labeled everything in the same. Only 20% of the carbon sources, both carbon sources, are labeled. Okay, but they are fully labeled. Okay, and then we could measure the labeling patterns in the amino acids. Okay. Which is protein-bound amino acids, where you can then infer from the amino acids, you can infer the labeling patterns in the metabolites, in the central carbon metabolites. Okay, so because their positions are conserved. Okay, so with this method, we could study very different. We could generate a great range of flux maps, okay, under different growth conditions to test our methodology. So we test glucose, glycerol, and plus methanol and glucose or glycerol, okay. Here are just some highlights of things we found out, okay. So for instance, first, of course, we had to define conditions where both substrates were co-assimilated, okay. So this means carbon limiting, where you can de-repress methanol assimilation, so that was one of the conditions. And then we could see how, for instance, the split ratio between methanol assimilatory and disammylate oxidation to CO2 was really sensitive to growth rates, for instance. So, for instance, under low growth rate, maybe you have between 50 to 70% of methanol goes to CO2, whereas at very higher growth rate, you have almost all methanol, it's oxidized to CO2, for instance. But you could also see that the big impact of methanol on glucose assimilation, so particularly in the split ratio between glycolysis and pentose phosphate pathway. So for instance, with methanol, more glucose goes to the pentose phosphate pathway, so generating, but no, you don't get more biomass, so it means you get a sort of an NADPH surplus there. And then also we found also a big impact on glycerol assimilation, particularly in the split ratio between gluconeogenic assimilation of glycerol or, let's say, catabolism to TCA cycles. So for instance, with methanol, the amount of the fraction of glycerol going to the TCA cycle was reduced, okay? So here I'm going to show you two or three examples we've done over the years on the use of the application of these techniques to different problems, okay? And the learnings we learn from that in terms of strain engineering and process development, okay? The first one was actually work done with, now I realize that the Boku logo, it's out. Okay, I need to update that. Okay, so so the first work, it was done quite a while ago. It was we were interested in the impact of oxygen availability on the physiology of Pichia, using glucose as a carbon source. So we use a gap promoter to express an antibody fragment, and we realized that, to our surprise, that when we were going to hypoxic conditions, the specific productivity was more than doubled. So we got an improvement on QP, like more than 2.5. Whoops. Okay. 2.5-fold increase. So we did multi-level analysis there on using DNA arrays, proteomics, and metabolic flux analysis, and then we discovered that under hypoxia, so glycolysis is transcriptionally regulated. Okay, so under hypoxic conditions, all glycolysis is upregulated, which is very different what happens in Saccharomyces cerevisiae, which is more, the regulation is more in the metabolic level. Okay, so no wonder that we got improvement in protein production because we were using a glycolytic promoter. Okay, but we then again went to generate flux maps on this situation, on these different states, and we could verify that indeed under hypoxic conditions that the glycolytic flux increased a lot, okay? And whereas, for instance, non-oxidative pentosulfate pathways was decreased, and then, okay, so also you have the decrease on TCA cycle, both at transcriptomic and metabolic flux levels, okay? So you had a nice correlation between these two levels, and also proteomic level. Okay, so so even though we didn't find any difference between the control strain and the producing strain in terms of metabolic fluxes, okay, we discover actually this big impact on both transcriptional and flux distribution. Okay, because of the oxygen availability. Okay. Very interesting. What we made up some calculations out of the metabolic fluxes, and we realized that even though under hypoxia you produce some fermentation products as redox zinc, we still had an imbalance, higher, let's say, an imbalance NADH generation. So there was, the balance didn't match. So we had an NADH accumulation there. So one of my PhD students had the idea, how about if using this NADH to do protein, okay, to channel this into anabolism. So what we did at the time, looking at the literature, we found out this an enzyme that actually catalyzes this direct conversion from NADH to NADPH. Okay, so it's from Saccharomyces. Pichia has an homolog, but it's not well characterized. So we just picked this gene from Saccharomyces. It codifies for an NADH kinase. It's a mitochondrial enzyme, but we remove the the targeting peptide. Okay, so we also express it in the cytosol. And when we found what was the following, okay, so we overexpress POS5 in the fat producing, in the recombinant strain. The mitochondrial overexpression didn't work. Okay, we didn't see any difference in production, but in the cytosolic overexpression of POS5, we could see that some of the clones were like producing 2.6-fold higher fat production. Okay, and we realized that those clones were actually harboring 2 copies of the POS5 gene. Okay. Okay, so if the hypothesis of my student was correct, this overexpression should be even higher under hypoxic conditions where you have a surplus of NADH. Okay, so we tried that. Okay, so we took these strains, the control strain with just fat production, 1 copy of POS5, 2 copies. Okay, and we did perform again chemostat cultures. Okay, so what you see here, it's in the middle, you have the the three strains under normal availability, let's say normoxia, okay, where you have this increase. The red one, it's 2 copies over the green, so you have this 2.5, 2.6 increase. But then if you go to hypoxia, okay, you have this synergistic effect. So you go you actually multiply, you increase 8-fold. If you add this post-5 overexpression plus hypoxia, you go up to 8-fold increase in specific productivity, okay, just with one genetic modification. Okay, so that was an example of what can you learn from metabolic flux analysis. Okay, this nicely correlated with actually the NADPH NAT NAP ratio in the cell. So you could see that for the strain with 2 copies, that this ratio was always higher, independently of the condition. The other example, okay. So the other example, it was with expressing of lipase from a fungus in Pichia using the AOX1 promoter. Okay, we knew beforehand that this enzyme, it's hard to express in Pichia. So actually, when you overexpress it, growth, the biomass yield goes down, and growth rate also it's decreased. So there's something going on with metabolism. Okay, so that was it. So we went up and tried to do this labeling experiments using a glucose methanol mix. Okay, we use, I will skip that. We used 2 different strategies. I will skip that. And essentially, we perform a metabolic flux analysis using 1 control strain. Okay, with no lipase production, and 2 independent clones. Okay, and from the macroscopic data, you can already see that there's big differences. Okay, significant differences, I would say, between the control and the producing strains. And for instance, the substrate glucose and methanol substrate uptake rate are increased in the production strains. You have more CO2 produced in the raw producing strains, and also the biomass yield is slightly decreased. Okay, and this is surprising because we're producing very little protein, milligrams per liter, not grams. Okay, so doing metabolic flux analysis, we could see that with the producing strain, the glycolytic flux was increased. We also increased what would be the methanol. The methanol that it's directly oxidized to CO2, while the pentose phosphate pathway is reduced. So in a way, you're reducing the NADPH that's produced, and you're increasing the NADH. Okay. Also, the TCA cycle activity was reduced, and we could see also differences in what would be the redox shuttles, okay, across the mitochondrial membrane. So based on that, we try again. Okay, if we have a problem with limitation of NADPH, let's try again post-5. Okay, it worked a little bit. Okay, depending on the growth, we tested 2 different growth conditions with methanol or glucose, methanol and glycerol or glucose mixers. Okay, so in blue you have on the right-hand side you have increase in productivity, so you don't get big things with the blue. Okay, you have increase on between 20-40% overexpressing post-5, while we also tested another strategy that it was trying to counteract this excess of NADH. Okay, and so by introducing an alternative NADH oxidase. Okay, with the idea that this would try of enhance what would be the methanol assimilation and hopefully protein production. But the effect is actually very little, so it's maybe 10%, and it's not statistically significant. Okay, and the last 2 slides I have very quickly, if I may. Yeah. Okay. So it's applying the same tools to another topic, which is metabolite production in Pichia. So we have been working for the last few years in producing particularly 3-hydroxypropionic acid, okay, which is similar to lactic acid, okay, And also it's an interesting platform chemical. So we started with the idea of producing 3HP from glycerol, and this can be done very relatively easy by just overexpressing the malonyl-CoA reductase. So with one enzyme, you can actually go from malonyl. You can actually get from malonyl-CoA, you can get 3HP. Okay, it's very demanding in terms of NADPH. So that's why from the start we also thought on introducing POS5. And with this basic strain, we got quite decent values of 20 grams per liter of 3HP. Okay, although we also realized that there were some byproducts like erythritol identified in fed batch cultivations. We try then to do let's say the classical metabolic strain improvement, okay, essentially overexpressing or putting additional copies of the malonyl-CoA reductase, okay, so just try to pull down and get more product, and also pushing, so it enhancing or overexpressing genes upstream, okay, to produce more acetyl-CoA and then acetyl-CoA and malonyl-CoA, okay. And we got very, let's say, not disappointing, but puzzling results. So in some cases we were getting some improvements, okay, and in some other we were even going worse, okay. So even that we could, okay, enhance we could go from 24 grams per liter to 37 grams per liter and improve quite a lot the productivity. So we decided now to apply this metabolic flux analysis to really get insight on why we didn't get the expected results, okay? Because this was working, for instance, in Saccharomyces that were getting, when doing this modification, getting double productivity, etc. So here and not in Pichia, okay? So since I'm out of time, we just published a paper a few months ago on analyzing these 18 different strains we generated. Okay, of course we didn't do this in chemostat cultures. We do it in a high throughput manner in microbioreactors, and of course in batch mode, so in exponential phase. Okay, so from there you can get quite nice diagnosis of what is happening. Okay, we have some diagnosis, and then we have here new clues to make another round of metabolic engineering to improve, to release these bottlenecks, okay? And with this I will conclude now. So I hope I was able to show you that metabolic flux analysis based on carbon-13, it's a powerful tool to understand the impact of both environmental and genetic perturbations, but still there's a long way to go, okay? So there's difficulties in let's say, getting fluxes across different compartments, cell compartments in a very precise way. Metabolic flux analysis under non-stationary conditions, so meaning fed batch, etc. It's difficult, very demanding. Okay, and of course we would like to have fluxes outside beyond what would be the central carbon metabolism. So this is even more demanding. Okay, so just thank also all the PhD students that contributed to these studies, and of course all of the collaborations, people that help us with these techniques. Okay, thank you.
Pau, can you comment on just the scale that you do it, the labeling?
The labeling, yeah. Bioreactor, all the bioreactor work in chemostat, it's conventional. So it's 1 liter, so the working volume is 1 liter. Okay, so the labeling, it means, yeah, it's expensive, they're expensive. Of course, you would like to go to lower scale, okay? That's why for the last example, we run it in micro, 2 ml reactors. And there you can, it's very difficult to make a chemostat continuous cultivation at that scale. So that's batch cultivation. Okay. I must say that the results are not comparable because fluxes are very sensitive to growth rate. So in a batch, you measure flux rates at maximum growth rate, and then in chemostat you're limiting growth rate. So yeah, you will get different things.